Hi, I'm Dr. Sarah Beth Redmond, and today I'm going to talk about some ideas related to the biology of the undead. The concept of undead and zombification of animals has been studied a lot in particularly insects and fungi, but today I'm going to be applying these ideas to the potential for and reaction to the future in which there is some sort of zombie outbreak and need to respond to that from a biological perspective. So some basic principles I'm going to be following for combating a zombie outbreak scientifically kind of relate back into the scientific method. So I'm going to give some recommendations for observations, how you might build a model and a hypothesis about the zombie outbreak, how you would study the undead, and how you would eventually reach the point, hopefully, of experimenting on the undead and trying to find a cure for that. So the first part of science is always to observe. Sometimes this is observing the literature that's been published on a topic, but in this case, since there is not a whole lot of literature on zombies, we're going to be going to primary sources. This means you're going to need to have a way to watch and document the activities of zombies. This would recommendedly take place from a safe location. So I've recommended binoculars or another form of long distance observation for this so that you can get yourself to a high secure location and not be at risk from attack or other disturbance by the undead while you're doing this. It's important to take very detailed notes when you're making observations on something you don't understand, because you might not recognize initially what is important. This is true when you're learning any new thing, that as someone who's not really experienced or educated in that particular area, you might not right away identify what are the things that are going to be really crucial for you to understand that. So things like times, weather, number of undead, number and species of non-undead. These are all going to be relevant notes for you to take as far as your understanding. And it's also important to mention that after you observe, you need to take time on a regular basis to go back through your notes. You might not notice a pattern as you're taking notes, but as you go back through multiple days or weeks of information, things might jump out at you that you did not initially notice. So some things I would observe for, first is, how does zombification or undeadness spread? So some commonly portrayed ways that this can happen are through direct contact, like biting, touching, or skin puncture, environmental exposure, if you've gone near a certain location that's contaminated in some way. And then there's also some cases where we see corpse conversion, where living people are fine, but as soon as you're dead, you are likely to be converted into an undead individual. Another thing I would be really interested in straight away is which species are affected. And I've kind of given a hierarchy here. Humans only is often represented, but there are lots of things which affect humans, which would also affect primates, or might also affect mammals, or might affect all vertebrates, or we could even go broader than that. This is particularly important if there is actually some sort of undead apocalypse, because you're going to need something to eat, and you don't want to eat these organisms until you know that they aren't affected. Knowing that organisms are potentially a vector for transmitting the zombification to you means you probably shouldn't eat them. So these observations help us answer a couple questions. First, an important question is there some sort of infectious disease or chemical exposure that we could potentially prevent? If we have preventable means and we can identify what is causing this particular outbreak, then we're much more likely to be able to combat it. Secondly, we want to know what are the features of organisms which are affected. We know based on evolutionary history that there are a lot of similarities which are shared by each of these increasingly broad groups. But if we can identify that there is a point at which groups of organisms that share some features are not affected, then we can start to identify what features they do not share and hone in on those as potential reasons why they're not affected, which could impact development of a cure. There are many other observations you might want to record. I'd encourage you to think about those. Once you've made these observations, you're going to want to start to hypothesize about what is going on in this outbreak and how these undead are being generated. This is an example of a published model to explain zombification and how the pools of individuals shift between the happy, living, normal humans that go through a process of zombification or through natural death, and then how the potential for reanimation of dead zombies might also cycle dead individuals and zombie individuals. So this is just an example. There might be other schemes depending on the mode of zombification overall. This is important because your model is going to determine what you can expect to achieve through treatment. So if you think about each of those processes as having a rate of individuals being affected, then you can start to calculate based on a population value, which we know, the rate at which we can expect people, these susceptibles, to be converted into zombies, shown in the red line here. And this is the example that was given by Smith et al. And it shows initially, if you look at these, it doesn't look that promising. The main thing is that we end up with some people that are alive at the end of this. But it's important to note that the x-axes are not the same. And if you stretch them out to be at least similar, then we have a longer period of time in which to identify the cause of this outbreak and start to combat it. And that's a really useful thing. Time is going to be of the essence in a zombie outbreak. So if you identify that treatment could stop the epidemic, you need to move on to studying how to do it. Some precautions. A lot of the potential causes of an undead outbreak are going to be things that would be transmitted through contact, or at least could be transmitted through contact. So you want to protect yourself. Gloves are going to be a great thing to have. But also you want to be able to sterilize any biological contaminants. 70% ethanol is the go-to for this kind of basic level of sterilization. There are some higher levels, but this is a good precaution. So if you can, if nothing else, find a liquor store and get some vodka, and that should work just fine. So some things you might also want. A source of zombified tissues, especially surface tissues, will be useful because they're the most easily accessible. Some stains would be useful, and a microscope if you can get your hands on one. Find a kid's toy store or a university lab and get your hands on a basic compound microscope. So comparisons that you might make in a study to find out what is affecting the undead. For the purposes of your study, you are the most reliable sample of a normal individual. So you're going to want to look at how your biological structures and behaviors match up to those of zombies. So you might think about cellular structures. This is a picture showing a microscope image of stained human tissue, looking at skin layers and then the layers underneath the skin. So this is a normal image. You can see that there's lots of organization. Most of the edges line up very nicely with the edges of cells around them. And we've got very distinct zones. Some questions you might look for that relate to specific cells and specific organization. First, is the immune system responding? If it is, then that gives the suggestion that this might be an infectious pathogen that's causing the outbreak, and you could then start to plan in that regard. All these cells that would be found in the blood are going to with some fairly normal stains that you would find in pretty much any university biology lab, stain very dark purple and show lots of variation in their size and their granules, are part of the immune response. So those are good things to look for. You can also look for in your tissues whether the blood is circulating or whether it's left circulation. So here you can see a blood vessel with lots of nice layers surrounding it so that it can constrict. But you can also see that there's a lot of these little donut-looking red blood cells outside of the blood vessel. That's not a good sign. That means that blood is not behaving normally, and so we might have some sort of problem. If you identify this in many locations, then that could be an issue. I'll also point out that these darker, kind of purpley black cells are some of those immune cells I talked about in the last slide. And then this is a really catastrophic thing that you might encounter where we've got necrosis of a lot of layers. You can see that the organization of cells has changed a lot. There's many, many red blood cells outside of tissues. There's a lot of white blood cells, these purple stained nodes that are in this area. This is a sign that there's a lot of damage that the body's trying to respond to. I'll also point out that it is possible that the immune response might not respond in a zombie. We don't know. Lastly, something you could look for microscopically is the presence of a foreign cell type. So fungal, protozoan, and bacterial cells would be visible to some extent as something present in an undead specimen and not in a live specimen. The unfortunate thing, because we often represent zombie viruses, they would not be visible under a normal light microscope. The response, especially if there is an immune response, would be visible, but the virus itself is not something you would see unless you know how to operate an electron microscope. You might also make behavioral observations. Looking for sensory function and reaction time are good things to consider. The behavioral observations are going to rely on measure whether or not the neurons are functioning actively in these undead. There are two important parts to neuron function, and these are both the parts that are really critical to maintain in order to have a neuron that can respond and learn. And those are the action potential, for which we need to have good membrane integrity, so the membrane has to be maintained. It can't be getting broken down. And then also control of ion movement. That has to do with both protein production and the availability of ions. Normally we resupply them from our diet, and our renal system maintains normal levels. We're also going to need neurotransmitters, which are primarily related to our protein reserve. So especially if the undead aren't able to eat and digest, then they would not have a good supply of neurotransmitters. And then we're going to consider some levels of brain function as they would be reflected by behavior. The caveat is that we don't have a whole lot of opportunities to study these responses independently, so this is with a grain of salt. Your basic hindbrain would be responsible for breathing. Even if the zombie doesn't necessarily have to breathe, it might still continue to perform that function with its diaphragm, as well as swallowing and visual tracking, following an object. The amygdala, if it's functional, then we'll have a fear response. If the occipital lobe is functional, then we can discriminate between humans and zombies. And if the hippocampus is functional, then we can mimic or follow procedural routines. And if there is full cerebral or prefrontal cortex activity, then we can follow a schedule or a hierarchy, as well as show emotion. If any of those things are possible, then we might want to consider our treatment plans. A vaccine would prevent treated individuals from undergoing zombification. And there are some different strategies we might take for generating a vaccine. An antimicrobial antiviral drug would kill the pathogen or neutralize a chemical. or we might actually be able to cure and reverse the zombification process. This would take a lot of experimentation to determine whether it was possible, but it would be the most optimistic outcome biologically.